us to lead us in a short word of prayer before we can begin, so that we can have good network and we can have a constructive discussion, as well as be able to learn from today's discussion. So who wants to volunteer to pray for this session? Can we have volunteers? Yes, Mukisa Sandra.
Precious Father, in the name of Jesus, we want to thank you for this day. We want to thank you for that precious gift of life. Father, we dedicate our discussion unto your hands. May you give us the gift of knowledge, wisdom, and understanding. May you guide us through with our teacher, Lord. And may you help us understand, Father, King of Glory. We ask you to help us with the gift of good network and understanding. In the mighty name of Jesus Christ, our Lord, we are prayed and believed. Amen.
Amen. Thank you, Sandra. Yeah, so I have picked a topic that I think we have all covered, regardless of the different levels of coverage. It's an S5 topic, but that usually disturbs the candidate. So we are going to look at the topic of photosynthesis, and we are going to have a number of questions we are going to discuss. And I hope to have a very interactive discussion with each one of you on this call. So here is our first question. Describe chemosynthesis in nitrifying bacteria. How do the nitrifying bacteria carry out chemosynthesis? I'm sure you've come across this term, chemosynthesis, and we know that it occurs in the nitrifying bacteria. So how do they carry out chemosynthesis? To start with, what is chemosynthesis? Does anyone want to share with us? Galaxy A12, I can see you've unmuted. You can kindly tell us what chemosynthesis is. Galaxy A12. Okay, anyone who is ready? What is chemosynthesis? I was thinking that this is a very friendly topic. Perhaps I was not as mistaken. So when we talk about chemosynthesis, yes, Mukisa, Sandra.
So teacher, for me, I think chemosynthesis, this is the process by which chemosynthetic bacteria synthesize organic materials from inorganic materials through the use of special chemical processes instead of sunlight.
Okay, thank you, Mukisa Sandra. Yeah, so like she has said, it occurs in chemosynthetic bacteria, for example, the nitrifying bacteria and the nitrogen cycle. And it's a special type of autotrophism, except that here, the organisms use the energy from the chemical reactions to form their organic compounds. Unlike in photosynthesis, where they use the energy from sunlight to convert inorganic compounds into organic compounds. So we want to discuss the stages that these nitrifying bacteria utilize, or the steps in order to make their own food by chemosynthesis. So what are the step-by-step reactions that take place when the nitrifying bacteria are converting the inorganic compounds at their disposal, okay, into organic molecules? Can we at least try? Assuming you have found this question in your paper in November, what would you write? This is very quiet. Okay. So this is what I have for us. So first of all, we know that when dead organisms are broken down, okay, these dead organisms contain, once they were living, they had proteins in them, and these proteins are broken down to form ammonia. That's why when you pass by a rubbish pit, there is a very pungent smell. So the ammonia is given off in the process of breakdown of plant and animal proteins. So this ammonia enters the soil and it combines with carbon dioxide, and then we are going to form ammonium carbonate. So this ammonium carbonate is oxidized to form nitrous acid. So the process of doing that will generate energy, and we shall see that this energy is going to be put to some further use. So the organisms responsible for this are the Nitrosomonas and the Nitrococcus. Remember, both of them are nitrifying bacteria. They are converting the unusable form of nitrogenous compounds into a usable form. Okay, remember we have from ammonium compounds, then we have nitrites, then we have nitrates, and then the plants can eventually absorb the nitrates because the other forms are unusable and some of them are actually very toxic. If you have been very observant, when you put fresh cow dung on some plants, the plants instead get, the reaction is so bad that sometimes the plants fail. But if you got cow dung that has decomposed over a period of time, and those toxic compounds have been broken down to less toxic ones, then the plant will do well. Okay, so then the nitrous acid combines with salts in the soil. For example, we may have magnesium or calcium salts, and then they will form nitrites, and then the nitrites will form the nitrates. And this is done by the bacteria, and in the process, also energy is given off. Now, if you look at this second step, you remember when an acid reacts with a metal, okay, a metal substance, then it can give you a metal salt, okay, and water. That's like a bit of chemistry. Then, of course, like I said, the energy is now utilized to synthesize the organic compounds. So if you remember what Sandra told us at the beginning, that these organisms utilize the energy from chemical reactions in order to make organic compounds. So those are the chemical reactions that are involved, and that is what the energy is used for. Okay, so that was our first question. So if you have any queries, you may ask, or you may take some screenshots before we can go to another question. Okay, so the silence means yes, Sandra.
Teacher, so this question majorly wanted us to describe how we obtain energy from the chemical reactions to be utilized in chemosynthesis.
Yes. So they wanted you to talk about what kind of raw materials those bacteria use to generate the energy to synthesize their organic compounds. So remember, these bacteria are very important in the nitrogen cycle, in the making nitrogenous compounds available to plants in the soil. So this takes place in a number of steps as we have observed.
Okay, thank you.
Okay, let's go to the next. This one. Okay, this one I did not hide the answers, so it's not going to help us much. But account for the coexistence of purple sulfur bacteria in weeds or ponds and rocks. So I didn't hide these answers, so I think we can just try to understand what the answers are. So I hope we all remember the purple sulfur bacteria. And the weeds. You know that the purple sulfur bacteria rely on light energy to make their own food, and so do the weeds. So it would appear like they are going to compete for the same amount of light, okay, for the same type of light. But yet we are told they can coexist. And from our ecology, we know no two species can occupy the same ecological niche, okay? They can't occupy the same habitat unless they're occupying different ecological niches. So we see here that their requirements for survival are different. So in that case, they cannot be in competition. It is like if you have a zebra, maybe that feeds on tender grass, and you have a buffalo that feeds on coarse grass. Coarse grass, I mean the grass that is very hard. So if they are grazing together, we cannot say that they are competing with each other for the same food. The zebra will eat its softer grass, while the buffalo will look for the harder grass. So in the same way, we find that these two organisms are coexisting because the light they are interested in, or the light they are able to absorb to allow them to make their own food, lies in different spectra, okay, in the different regions of the white light spectrum. So you find that the purple sulfur bacteria are going to absorb, okay, light in the ultraviolet range, okay, and in the infrared range. Now, these two forms of light lie outside the part of light that the weeds would need. So it means that the light for the weeds will filter through and not be utilized by these purple sulfur bacteria because they don't have the right pigments to absorb it. Get it? And then they will be able to make their own food just like the purple sulfur bacteria are able to make their own food. So I hope this is clear to us. Okay, I think we are going to proceed if there's no further question. Enos, you want to tell us something? Okay, I think we shall proceed. So we have another question here, which is telling us to explain how ATP is synthesized by the chemiosmosis in the chloroplast. I think we've heard of chemiosmosis before. So how does chemiosmosis bring about ATP synthesis? Yes, Enos. If you have something to say, please, you can also share in the chat in case you have a problem with your mic so that we can be able to appreciate what it is that you want to share with us. So I have an illustration here, which is showing us the thylakoid membrane. When you look at the thylakoid membrane, it is actually a phospholipid bilayer. Remember, all membranes have phospholipid layers. So when we look at the thylakoid, there is what we call the thylakoid space. That is between the two parts of the thylakoid, the outer membrane and the inner membrane. Maybe I can try to illustrate this. Just a moment. Let's annotate. So assuming we have something like this and something like this. So on this side, we have the stroma, and on this other side, we have the stroma. So what you're seeing right here is like shown by this one line here. And then there's a similar thing on the other side, which is shown by this line. So this other membrane is not indicated. I'm only indicating one side of the membrane, okay? And so this space in between these two membranes is what we are calling the thylakoid interior space, okay? So along the thylakoid, you will notice that there are a series of electron carriers, okay? And then we have also photosystems, and then we may also have ATP synthesizing systems, what we call the ATP synthetase, okay? So what happens is that when this plant receives sunlight, okay? When it gets sunlight, it is going to provide sufficient energy to split the water molecule, okay? What we are saying here. So when water molecule is split, it is going to form electrons, it's going to form oxygen, and it's going to form also some protons, okay? So when it forms these three substances, so the protons are going to be exchanged by what we call the electron transport chain or the electron transport system. So they are moving from one electron acceptor to another electron acceptor, okay? And we are seeing that while that is happening, the hydrogen ions are going to be pumped into this space. So they are pumped into this space, which I have put right here. So when I say they are pumped, it means that there is active transportation, okay? So they are pumped inside there. That means they are being taken against the concentration gradient. So when they move against the concentration gradient, it means that they are going to accumulate in this the thylakoid interior space. So when they accumulate there, then they are going to generate sufficient potential energy. So because now we have established a steep concentration gradient of hydrogen ions between the interior space here, the thylakoid interior space, and then the stroma right here, they are going to escape, because remember what I didn't mention earlier is that this membrane, this phospholipid bilayer here, is impermeable to these hydrogen ions. So they cannot diffuse out because the membrane is selectively permeable to them. So it does not allow them to come back down the concentration gradient. So they'll accumulate in this space, and when they accumulate in the space, then they will get a way of escape through this ATP synthase, okay? So when they reach here, remember they will have accumulated and they have a high potential energy. So they are going to come down the concentration gradient by diffusion. And as they do that, the energy generated as they are coming through this ATP synthase is going to cause the combining of ADP and the inorganic phosphate, and that will give us an ATP. In the meantime, these electrons that were being exchanged, they were being moved down the electrochemical gradient, okay? So when electrons move from a high electrochemical position and they come to a lower one, what happens is that a lot of energy is given off. If you remember from your chemistry, when electrons are coming to a lower energy level, they emit energy, okay? And so when that happens, it's going to come and cause the formation of this reduced NADP, okay? Reduced NADP, of course, in combination also with the hydrogen ions that have come through, because remember there were hydrogen ions here, they have combined with rather the hydrogen ions have come through here, and then now, yes, they're going to combine with the NADP, which has also accepted the electron. So you get what we call reduced NADP. Yeah. Someone says they are not hearing properly. Maybe you can increase the volume of your mic so that you can hear properly. I don't know if now I am louder than I was earlier. I hope I'm louder this time. Okay, it seems my voice, I was speaking a bit far from the mic. So I hope we have followed this process of chemiosmosis. So it is outlined right here, but I was trying to explain to you how it takes place according to this drawing. So this chemiosmosis process results in the formation of two useful products, that is reduced NADP and ATP. So this chemiosmosis process is the same as the photophosphorylation, except that here we are trying to emphasize how the thing exactly happens, okay? But the Z scheme and the other things in the books, they are more simplified versions of what we are showing here, except that they don't emphasize the chemiosmosis aspect of what takes place. So this and what you already probably learned about in photophosphorylation, cyclic and uncyclic, they are actually one and the same, but this one gives you more details. But you answer this question the way we have done it here if the question talks about chemiosmosis. If it doesn't talk about chemiosmosis, then you can stick to the other way of explaining how photophosphorylation occurs. Remember, photophosphorylation means the addition of an inorganic phosphate to ADP to form ATP in the presence of sunlight. So we have photophosphorylation and we have other forms of phosphorylation. So when it's done in the presence of light, then we call it photophosphorylation. But if it's done without light or without a requirement of light, as we see in respiration, what we call oxidative phosphorylation, then we don't call it photophosphorylation. So I hope this is clear. You could take some screenshots. If there's something that's not clear on this explanation, you can seek clarity so that we can all move together so that nobody is left behind. So are we together this far?
Okay, excuse me, sir.
Yes, please.
I just wanted to know, from the diagram, we can see that photolysis of water is taking place in the thylakoid lumen. Yes?
No, actually, it's not taking place in the thylakoid lumen. Okay? It's just that the illustration couldn't bring it out. Okay? It takes place on the lumen, but I don't have three-dimensional way of illustrating this. You get it?
So where exactly is it taking place?
It takes place on the surface.
Just on the surface?
Yes. So remember, the water has been absorbed and then the leaf is exposed to sunlight. So that light has sufficient energy to split the water, what we call photolysis. And then the products of photolysis undergo the stages as we have shown. So the electrons are going to be picked up by the electron acceptors, okay? Yeah. And then the hydrogen ions that are formed are going to be now pumped into the membrane, the thylakoid space. From outside, if you look at the arrow, of course, the oxygen will diffuse out of the leaf and go into the atmosphere. I didn't have enough time. I'd wanted to get a video showing this very exact process. But since you have some data, you can go to YouTube and get a video on chemiosmosis. They will show you this very process, how it happens step by step.
Okay, sir.
Yeah. Any other question? Okay. If there isn't, then I think we need to proceed.
Another thing, sir, Shutu.
Yes.
Hello. I would like to know whether there is a significant process how the ATPase is manufacturing ATP from the energy.
Now, the ATPase is a complex. It's a complex. And what happens, if you remember, enzymes provide, lower the activation energy for chemical reactions to take place.
Yeah.
Yeah, so what happens is that as the hydrogen ions are coming down, imagine when you have a tank on top of your house, okay? And then it's full of water. and the water and the tank, say, gets a big crack on it, the force with which the water comes down is very great. So that amount of energy involved as these things are coming down is what causes the formation of that bond between ADP and inorganic phosphate. Because the higher you go, if you had a storage building and you had a tank, even if you had something as small as a nut falling from a 10-story building on the ground, and then you have another nut falling from a 2-story building, the force with which that nut hits the ground will not be the same. You get it?
Yes, sir.
Yeah, so the fact that we have accumulated so much hydrogen ions in that space, and then suddenly you are releasing them to go out down the diffusion gradient, that force is so great. So that energy is sufficient to form the ATP molecule. All right, so we are going to move on. Oh. So we also have another question here, that although photosynthesis generates ATP, as we have observed, plants also generate ATP by respiration. So we are wondering why the plants bother themselves to generate ATP by respiration, yet they can also generate it by photosynthesis. Why they can also, yet they can also generate it by photosynthesis. So from what we can see, I hope we remember the Calvin cycle. So in the Calvin cycle, let me try to annotate. So in the Calvin cycle, remember, we start with rubip. So you have maybe a rubip. Okay, let me. Okay, right here you have possibly carbon dioxide combining with rubip. So I'm going to ask, when carbon dioxide combines with rubip, what do we form? Then you have carbon dioxide here. Yes, if you know the answer, you can form it in the chat. Okay, Sandra says GP, another Michelle says PGA. So it's better we say these words in their fullness because sometimes they can be confusing. So when carbon dioxide combines with rubip, remember rubip is a 5-carbon compound, and then carbon dioxide is a 1-carbon compound. So you're going to get a 6-carbon compound. However, we know that that 6-carbon compound is unstable, okay? It is unstable, so it immediately degrades or breaks down to form 2 molecules of 3-carbon compounds. So those 2 molecules of 3-carbon compounds are phosphoglyceric acid, or what some people have called phosphoglycerate. So we have phosphoglyceric acid or glycerate 3-phosphate. So all those are different ways of saying it, okay? Phosphoglyceric acid or phosphoglycerate glycerate or glycerate 3-phosphate. Now, the glycerate 3-phosphate, as you can remember, is going to be reduced. It's going to be reduced by adding reduced NADP, which will give up its hydrogen ions, and then the ATP, which will give up its phosphate. So we are going to form another molecule, which in this case is what we call glyceraldehyde. Glyceraldehyde. What some books call the triose phosphate. The triose phosphate. So the triose phosphate partly is used to form hexose sugars. It is used to form hexose sugars. Please, can we mute our mics? It used to form hexose sugars. And however, that is a small fraction of the sugar, whatever the triose phosphate or the glyceraldehyde phosphate. So the majority or the most of it is used to regenerate the RuBip. So, but the process of regenerating RuBip utilizes most of the ATP. So you find when most of the ATP has been used, very little ATP is left. So the plant cannot rely on very little ATP that is generated. So it means the plant has to still carry out respiration. And from respiration, as you can remember, it can generate a lot more ATP than probably photosynthesis would have made. Because remember, if there is aerobic respiration, we are going to have ATP from glycolysis, ATP from Krebs cycle, ATP from the electron transport system. And when you combine all those different processes, they are going to generate a lot of ATP. So that is why in this question, we say that most of the ATP produced during light-dependent stage of photosynthesis, it is utilized to regenerate the RuBip from the triose phosphate, okay? And then it will form what we call phosphoenolpyruvate, okay? From the pyruvate, and then of course that one will be used to form the RuBip, and then, yeah. So there's very little that is left. Someone is saying that they think that the ATP from the chloroplast cannot cross to the mitochondria. Well, that is quite a good thought. I cannot comment for sure whether that is the case, but assuming it was relevant for the plant, then maybe there would be a way for that to happen. But for now, we can only speculate. Okay. So we are going to go to the next question, hoping that we have all understood this one. So can we describe the similarities between photosynthesis and anaerobic respiration? I expect an answer from at least each one of us. I'm going to start calling names. Daniele, can you give us an answer? In what ways photosynthesis is similar to anaerobic respiration? So someone tells us they both utilize carbon dioxide. Does anaerobic respiration utilize carbon dioxide? Actually, the word should have been aerobic, not anaerobic. Sorry about that. This should be aerobic. No wonder some people are getting disturbed. So both form ATP. That's true. In both processes, ATP is formed. So when I was typing, I was in a hurry. So I didn't edit some of the things. So let's do the need. Let's do the needful. Okay. Someone says they both involve enzymes. That's true. Both of them occur in cell organelles. Okay, they all occur in the same cell organelles. Both are enzyme controlled. That's also true. What else can we think about? Angel? Okay. The rest of us are quiet. So both of them are energy converting processes. So in photosynthesis, we are converting light energy into chemical energy. In aerobic respiration, we are converting energy from the food molecules, okay, into ATP. And then that ATP will later on be, of course, used to generate energy for the different processes. Both take place in the living cell, true. They both undergo the electron transport chain. Both of them use ATP. Okay, that's also correct. What have I missed out? Both spend energy ATP in both chemiosmosis occurs. Both require ATP. Okay, all of these submissions are fine. Then we have involving enzymes. Someone said that. Occur in living cells. Someone mentioned that already. Flow of electrons. Someone has mentioned it when talking about chemiosmosis. That one is covered. Require special organelles. Okay, I think someone said they occur in organelles. Wanted probably mean this. So they all don't take place just anywhere. They take place on special organelles. And then this is just emphasizing that earlier point, chloroplasts for photosynthesis and mitochondria for aerobic respiration. Then they require mechanism of exchange of gases. So in photosynthesis, there is an intake of carbon dioxide and an outtake of oxygen, whereas in aerobic respiration, there is an intake of oxygen and a giving off of carbon dioxide. So that's why we say they are both involving an exchange of gases, okay? So in both cases, gases are being exchanged, and because they are enzyme controlled, therefore they are going to be affected by the temperature. So if there's high temperature, then the reaction will slow down. If there's very low temperature, it will equally be slowed down, of course, for different reasons. At very low temperatures, enzymes are inactivated, but at high temperatures, they are denatured. And then we talked about ATP being formed in each one of them. So I think we have done a good job there, okay? Someone says they both have cyclic reactions. Okay, that's an interesting one. You have the Calvin cycle for one of them. And then for the other, we have the Krebs cycle. So that's a good observation, Grace. Then we can go to the next question. So what are the differences? Because the next question is about differences. So what are the differences between photosynthesis and aerobic respiration? Let's get the answers coming through. What are the differences between photosynthesis and aerobic respiration? Are they the same in all ways? Can we have some answers?
Okay, so mine was that aerobic respiration occurs in both plants and animals, whereas photosynthesis occurs specifically in plants.
Okay, that is a difference. So photosynthesis, you could say, doesn't occur in animals, okay? Whereas respiration occurs in both plants and animals. Okay, that's right. Can we get another? Michelle tells us photosynthesis occurs in chloroplasts, aerobic respiration occurs in the mitochondria. That's also correct. Okay, what else? Brennan, you want to tell us something? Okay, let's hear from Daniele. Daniele tells us that carbon dioxide is used in photosynthesis. Okay, let me see what was the other part. While it is released in aerobic respiration. Now, when you are discussing differences, you need to be very, very careful. You need to have what we call a criteria for your differences. You can start by saying, if you had a table, you have 3 columns. In the first column, you write the word criteria. Okay, this is for purposes of revision. And then in the next column, you write photosynthesis. And in the last column, you write aerobic respiration. So you ask yourself, what is my basis of deriving a difference? So like one of us has already said, organelle, where it takes place. So you say organelle, where it takes place. Then you can say chloroplast, then mitochondria. Then you can also say gases that are needed, okay? Then you say carbon dioxide. Then the other side, carbon dioxide is not needed. You get it? Or you say oxygen is needed. Then you come to gases given off. Then you say oxygen is released by photosynthesis. The other side, oxygen is not released, or you can say carbon dioxide is released. Now, why am I saying all this? Because Daniele is talking about use and release in one answer. So you find her answers, her statements are correct, but they will not earn her a mark because she's comparing carbon dioxide, comparing the use, like raw materials and waste materials, all in one difference. So that is why you need to be very careful. So if you talk about the raw materials, talk about only raw materials. The byproducts or the waste materials, talk about them alone, okay? Compare tomatoes and tomatoes, okay? Or colors and colors, and then size will come by itself. I hope I'm understood, although I've tried to illustrate it with different concepts. Then someone tells us lactic acid is produced in aerobic. Oh, Quinto, we changed the question to aerobic respiration, not anaerobic, okay? So we are discussing aerobic respiration, not anaerobic. And then we have Grace telling us oxygen is the final electron acceptor in aerobic respiration, when photosynthesis, it is a byproduct, okay? So here we are talking about the role of oxygen, okay? Still, this is related to the earlier concept I was trying to explain to Daniele. You need to be extremely careful. Photosynthesis forms organic substances, while aerobic respiration, organic substances are oxidized. Yeah, so one of them is the anabolic, like Mukisa has put it, and then the aerobic respiration is catabolic. So it results in the breakdown of organic substances. If you can recall from your Krebs cycle, you will find that at the entry stage of the Krebs cycle, you have a 6-carbon compound. This 6-carbon compound is citric acid. When oxaloacetic acid combines with acetyl coenzyme A to form a 6-carbon compound, then progressively we see the 6-carbon compound being degraded. And at each stage we are forming carbon dioxide. So from a 6-carbon compound to a 5-carbon compound, then to a 4-carbon compound, okay? So, and at each time when there's degradation, carbon dioxide is given off. So that's why we say it is catabolic. It keeps on reducing. Then we have other messages. Okay, someone has mentioned it, Faisal. So let's continue and see what we might have left out. Let us see what people left out. So this is what I had for us. The most obvious answer, most of us left it. I don't know, it's usually very interesting that candidates try to look for complicated answers and leave the obvious. My number one is the most obvious, that photosynthesis only occurs in the presence of sunlight. Remember, sunlight energy is needed to cause photolysis of water and also cause the promotion of those, rather the oxidation of the photosystems. So without sunlight, then it means photosynthesis won't take place. The aerobic respiration, on the other hand, can take place whether there's sunlight or there isn't sunlight. So it takes place at all times. Then we see carbon dioxide and water used up. Here, carbon dioxide and water are released. You notice the way I put this second difference? I'm looking at just the same thing, the role of carbon dioxide and water, okay? In the second one, third one, I'm looking at the role of oxygen, okay? So I'm not talking about oxygen being released, then the other side I say carbon dioxide being released. That would not be proper. Then we also see it occurs, okay, the fourth one was already given to us. There's one which we have somehow mentioned, but not quite elaborated. That is the second last. It results in increase in dry mass. That's what we mean by anabolic reactions. Anabolic reactions result in lengthening of a chain, if you remember from your chemistry, that you're building up complex compounds from simple compounds. So whereas in anaerobic respiration, we are breaking down a complex compound into simpler compounds. So that's why there's a loss in dry mass. Then of course, the last one was already provided for, okay? The last one was already provided for. Then of course, in red, we also have the different hydrogen carriers. In the photosynthesis, we have a NADP or reduced NADP. And then in respiration, we have NAD and FAD. So the electron carriers in this case are different. So you see the criteria here is, so the hydrogen carrier. The hydrogen carriers are being compared. So that is all for the differences. Now, we have a number of terms we have always come across. Compensation point, limiting factor, and photorespiration. Who can tell us what compensation point means? When we are discussing photosynthesis, we always talk about compensation point. Arafat, oh, okay, Patricia.
Okay, compensation point, this is the light intensity at which the rate of photosynthesis is equals to the rate of respiration.
Okay, that's correct. So it is that light intensity, okay? So what happens during that time? Can we add on some more? Okay, someone has posted something in the chat. So someone is taking us back, the overview about dry mass. When you talk about dry mass, we also have what we call fresh mass. Fresh mass is the weight of an organism. Okay? When you get an organism and you just weigh it the way it is, that is fresh mass. Then dry mass is when you dehydrate it. If you want to appreciate dry mass and fresh mass, just imagine you have freshly harvested beans from the garden, fresh beans, and then you sun dry them. So when you sun dry them and all the water has been removed, the weight of those beans is now the dry mass, okay? But the fresh mass incorporates also the water. So the fresh mass can be misleading if you want to know whether there has been growth, because the fresh mass depends on the amount of water in the soil. So if there's a lot of water in the soil, then that means that plant will have a lot of water in its tissues, and therefore it will have a greater fresh mass. But dry mass emphasizes how much food has actually been added. That's why when you're doing pyramids of what? Of biomass. No, actually not biomass. When you're doing pyramids of energy, okay? Pyramids of energy are able to take into consideration that amount of energy that has been put. The challenge with doing dry mass is that you must kill the organism. And yet with fresh mass, the organism can be alive and you keep on monitoring how much more mass it is adding. So that is why there's a difference. So whenever photosynthesis takes place, there is stored starch, okay? But remember, as the starch is being stored, even some water is present. So that is now the fresh mass of the organism. But if we were to remove the water and account for only the food that has been added, then that would be like the dry mass. Okay, hope that is clear. All right, now someone had just told us about what compensation point is, and we had agreed that her answer was correct. Now, Michelle is also adding to what we were told early. So what happens is that there is no net exchange. That's what I wanted us to talk about. No net exchange of gases. You find that exactly the amount of oxygen given off by photosynthesis is the exact amount of oxygen utilized by respiration, and the exact amount of carbon dioxide produced by respiration is the exact amount of carbon dioxide utilized by the photosynthetic process. So you find that no gases are actually going out of the plant. Neither oxygen escapes nor carbon dioxide, because the two processes are exactly balancing out each other. So basically, that is what the compensation point is. So thank you. Was it Opio or Lakelo? It's one of you. I didn't get the name right. Thank you for that. So what is the limit? What's meant by limiting factor? What is a limiting factor? What do we mean by limiting factor? Tito, please stop what you're doing. Yes, Sandra?
Teacher, a limiting factor is a factor that is in minimum amount and affects the rate of a chemical reaction in case it is affected by a number of factors.
Okay, you're correct, but you have not organized your answer well. You needed to have started with what you said last. Patricia?
Okay, a limiting factor. In a chemical reaction which is being affected by a series of factors, the factor which is in the smallest quantity that has a direct influence on the rate of the reaction, that when if its concentration changes, the rate of reaction also changes, is a limiting factor.
Okay, that is better put. So we start by acknowledging that some reactions are controlled by more than one factor. So the question now is, which factor is directly now affecting the rate of that reaction? So if that factor directly affects the rate of reaction, then we call that the limiting factor. So if you have many factors, now that particular factor that is at its minimum, okay, or at its lowest, there could be a concentration, but I'd rather say the factor that is at its minimum. That factor that's at its minimum will affect the rate of reaction. So when you increase that factor, the rate of reaction significantly increases, okay? So if you want to appreciate this, just imagine you are constructing a house. So if you're constructing the house, you must have a number of things. You need materials such as sand, cement, you need bricks, you need labor, you need water. Now, assuming you have all the bricks that you need to construct that house, all of them in one place, all the cement that you would have needed, you have all the sand that you would have needed, you have all the water that you have needed, then you have maybe 3 workers at your disposal. It means that the work will be done slowly because you only have 3 workers. But if you now bring 5 workers, then now it means that the work will move a little faster. If you bring 10 workers, it will move even faster. If you bring 100 workers, now it means that the workers are no longer the limiting factor because you have more than enough workers. Probably some of them will become redundant or idle, and they will have nothing to do because there is little work to be done, and yet there are very many workers. So that's what a limiting factor is all about. So we see this a lot when we discuss photosynthesis and when we discuss enzyme reactions. So in enzyme reactions, where the enzyme concentration, rather the substrate concentration is low, the rate of reaction will continue increasing as long as we are increasing the substrate concentration. But when we keep increasing the substrate concentration, then there will be a time when no matter how much substrate you add, the reaction can't go any faster. So the reaction will plateau, or it will level, or it will remain constant. So at that time, it means that all the enzymes are saturated. Their active sites are saturated. They are occupied by substrate. So it doesn't matter how much substrate you provide. So probably now you may need to provide more enzymes instead. So that is what a limiting factor is about. Okay. So for instance, here I've given an example of where there is complete darkness. Okay. It means that now light becomes the limiting factor. Okay. Even if you raise the temperature or provide a high carbon dioxide concentration. So the complete darkness will cause light to be the limiting factor. Of course, this knowledge of limiting factors has been used by man to improve crop production. I hope we have heard about greenhouse gases, rather greenhouses. Do we all know what greenhouses are? Do we know what greenhouses are? So a greenhouse, let me see what people are posting, okay? Eno says yes. So a greenhouse is like a tent of some kind, but where we are growing crops. And these crops are covered in, sometimes they can be made of glass, sometimes they may be made of certain materials that are as if canvas. So by growing crops in a greenhouse, you're trying to control the external factors that affect photosynthesis. So at night, these greenhouses, they are provided with light so that they can continue photosynthesizing. Then they always pump a lot of carbon dioxide in them. So that carbon dioxide is not a limiting factor. Then, of course, they provide heat in them so that even heat temperature is not a limiting factor. So you find that the rate of productivity in a greenhouse is very high. So they are not limited by any of those very important factors. So on a cloudy day, light becomes a limiting factor. Okay. In a forest, also light can be a limiting factor because of the canopy above the low-lying plants. So what is photorespiration? Who can tell us what photorespiration is?
Photorespiration is the combination of oxygen and RuBP in the presence of RuP carboxylase to form 1-GP and phosphoglycolate, where phosphoglycolate is oxidized to form water and carbon dioxide, hence no ATP is generated.
Okay, thank you, Joseph. Do you have any other submission? So let me ask, what causes photorespiration to take place? Under what conditions can photorespiration take place? And in what sort of plants does photorespiration occur?
Photorespiration occurs in C3 plants mainly. C3 plants, and it is in under oxygen partial pressures.
In C3 plants under what?
Under high oxygen partial pressures.
Is it just the high oxygen partial pressures? There's high temperatures. What else? At low carbon dioxide partial pressures, okay? All right, thank you, Joseph. So this is what I had for you. So it is a light-dependent oxidation of RuBP by oxygen. Remember, we have an enzyme called RuBP carboxylase oxidase, or Rubisco. So this enzyme has a higher affinity for oxygen than carbon dioxide. So remember, carbon dioxide is a limiting factor most times because in the atmosphere, there is only 0.03% carbon dioxide, and yet oxygen occupies up to 21%. So when the plant actively photosynthesizes due to high light intensity, okay, then it produces so much oxygen that now the concentration of oxygen is so high that RuBP carboxylase oxidase, instead of binding with carbon dioxide, it binds with oxygen, okay? What we call competitive inhibition. So when that happens, now we are going to have that glycerate 3-phosphate, okay? We are having one molecule. Because remember earlier I told you, when we have carbon dioxide and the RuBP, we are supposed to form a 6-carbon compound, which is unstable, which immediately breaks down to form 2 molecules of 3-carbon compounds, which we call glycerate 3-phosphate. So 2 molecules of glycerate 3-phosphate. But now here, what happens is that instead of forming 2 molecules of glycerate 3-phosphate, we are going to form one molecule of glycerol 3-phosphate, like Joseph has observed, and then another one called phosphoglycolate. Okay, so phosphoglycolate undergoes another series of reactions. Okay, instead of leading to the formation of triose phosphate, it will undergo a series of reactions. And in those series of reactions, they are going to use carbon. Okay, so we are going to have carbon dioxide being generated. You can imagine when we are carrying out photosynthesis, we are trying to add as many carbon molecules to build a very big compound of carbon. Now we are even using some of the little carbon that we have, and we are generating carbon dioxide, which means that this reaction is very wasteful. Okay, it is very wasteful. It is not helping the cause of anabolism or the cause of synthesizing large organic compounds. So that is why photorespiration is a very wasteful process for plants. And it's correct when you say it occurs in C3 plants. So C4 plants have overcome this process, and the CAM plants have also overcome this process. So maybe I will require you to talk about how the C4s overcome the process of photorespiration. Why is it that it never occurs in them? Okay, so let's go to the next. I want to explain why photorespiration is wasteful. I think I have labored to try and explain that, but let me see whether people have understood. Why is photorespiration said to be wasteful? Okay, someone is attempting in the chat. Okay, someone is suggesting that carbon dioxide concentration has increased to 0.04 due to climate change. Yes and no. It depends on where probably you are. If you are in a highly polluted environment, then carbon dioxide concentration will tend to be much higher. But of course, like we know, even a very high carbon dioxide concentration in that atmosphere doesn't help plants, because there's a limit within which plants can use the carbon dioxide. When it is too high, then it will interfere with the pH, and that will cause certain reactions not to take place. But otherwise, thank you for that input, Faisal. Sharon, can you tell us something? Michelle is saying carbon is lost in form of carbon dioxide. That's a correct observation, but you need to give me more than that. You need to give an explanation that is going to earn you more marks. Okay, if no one is responding, this is what I have. So the byproducts of photorespiration, which we call phosphoglycolate, okay? When it is oxidized, okay, we see that some of the carbon within itself is lost. And that's not the only problem. In the process of doing that, we are not generating any ATP or even any reduced NADP. So this in turn decreases the photosynthetic efficiency. Remember, we talk about efficiency. Efficiency is about how much you put out with whatever you have been given. Okay, that's when we say someone is efficient. Assuming you have been given 5,000 shillings, and then you use that money to buy many things. And another person is given the same money and they go to the market and buy nothing. They fail to get what to buy, or they buy just a few things. So it means that the other person has put their money to better use, because with little they have got so much. So you see that now with the photo respiration, we are wasting. Okay, so if we are to look at the amount of TP that is generated at the end, it is much less. Someone has put a comment in the chat that it is inefficient in that carbon, which is needed in building large molecules, are lost. Okay, that is also true, but you need to put in these other words, the glycolate, and talking about failure to generate ATP and the reduced NADP. All right, so how has sugarcane overcome photorespiration? Remember, sugarcane is one of the C3, rather C4 plants. Can we mention other C4 plants other than sugarcane? Okay, there is maize. That's correct. Is rice a C4 plant? Maybe millet. I'm not very certain about rice. We need to do some research on that. Barley, I'm also not very sure. Maybe sorghum. Okay, what was I about to say? Sim, I'm not very sure. We need to do research. Since we have some data, we can try and do the research about the C4 plants. Now, my question is, which parts of the world do we find C4 plants? In which parts of the world do we find C4 plants, and why? Maybe I can change that question. Tropical regions, that's correct. I would have said also, in which latitudes? Now, if we go to geography, within which latitudes do we find C4 plants? So those latitudes that are covering the tropical regions between, okay, someone has talked about altitude. Okay, there's also the altitude thing. There's also the latitude thing. The latitude is between the Tropic of Cancer and the Tropic of Capricorn. Then the altitude, of course, it is the low altitudes. And the reason is that at those tropical regions, we have high temperatures and we have high light intensity. Okay, so these plants are able to carry out photosynthesis without photorespiring, in spite of very high light intensities and high temperatures. Remember, those were the conditions that led to photorespiration. But there's something else that they do have that makes them overcome photorespiration. And that one we are going to see in our next submission. So we see that they have a special enzyme called PEP carboxylase. So in addition to the rubib carboxylase, they have PEP carboxylase. But then the other thing we know about them is that they have what we call the Kranz anatomy. What is the Kranz anatomy? What is the Kranz anatomy?
Okay, sir. Let me try to define. A crown section is a kind of arrangement of two concentric rings of cells around the vascular bundles in people plants.
Okay, thank you, Tamale. So you find they have the bundle sheath cells and the mesophyll cells surrounding the vesicular bundles. And because of that unique arrangement, you find that in one of those cells, only light stages of photosynthesis occurs, while in the other, only the dark stages of photosynthesis occur. So by so doing, you are separating rubic carboxylase from the light stages of photosynthesis, because the light stages of photosynthesis result into the production of oxygen, and then rubic carboxylase will react with the oxygen, bind with the oxygen, and then we shall have photorespiration. But when we have the different cells, the bundle sheath cell and the mesophyll cell, you're going to find that in one of the cells, the bundle sheath cell, which is one where there's the dark stages, you're going to find that you're having rubic carboxylase. But in the mesophyll cell, you have the PEP carboxylase, which has a unique ability to combine with carbon dioxide, even at very low concentration, okay? Even at very low concentration. So it will combine with and then form what we call oxaloacetate. The oxaloacetate or oxaloacetic acid will then be changed into malate. Then the malate will be shunted into the other cell, okay, and then it will be decarboxylated to form PEP, okay, and then the PEP will go back and be converted into actually to be decarboxylated to form a pyruvate. Then the pyruvate will be converted into PEP. So I hope that one is clear. So we are seeing here, one, they have a high affinity for carbon dioxide and does not combine with oxygen, this PEP carboxylase. Therefore, no photorespiration will occur. And then the PEP, the rubib, and the rubic carboxylase enzyme is found in the bundle sheath, which is isolated from the atmospheric oxygen, or rather from the oxygen inside the leaf, and preventing oxygen from reaching those cells where there is rubib. Okay, and then we see the carbon dioxide from the carboxylation of the malic acid or the malate accumulates into the bundle sheath cells, and it results into too much carbon dioxide being generated when we decarboxylate the malate. And then all this carbon dioxide can be fixed by the rubic carboxylase. So all of these points are important in helping understand how these sugar canes and other C4 plants can overcome photorespiration. Then, why is photosynthesis a two-stage process? We can go through that very quickly. Why photosynthesis is a two-stage process? We see that when we talk about photosynthesis, we say that one stage requires light and the other one doesn't require light. So if you provide alternate light and darkness, it will in turn affect how much food is formed by the plant. So which means that there are two stages, and one is affected by light and the other is affected by is not affected by light. Then we have the second point, that the rate of photosynthesis is influenced by both light intensity and temperature. So the light intensity, of course, affects mostly the light-dependent reactions, and then the temperature would affect the stages that require enzymes, mostly the dark stages of photosynthesis. So that in itself shows you if you provided low light intensity but high temperatures, then you would affect the rate of photosynthesis. And if you did the reverse and you provided maybe high light intensities but low temperatures, you'd still affect the process. Then if you provided both high light intensities and the suitable temperature, then the output would also be different. So we have another question on photosynthesis still. We can study this absorption spectrum for chlorophyll A and chlorophyll B, and also the action spectrum. So can we compare the action spectrum and absorption spectrum? Can I have some answers? I mean, there's so much quiet here. So how would we compare? What does the question require of us? Okay, similarities and differences. So what are the similarities? What are the similarities? Can you construct a more elaborate answer when you say both for peaks? I think we can do better than that. How can we best say that answer? Yes, both have 2 peaks. What else? It's true they both have 2 peaks. Is that all we can talk about the peaks? I think I need more than that. Someone tells us Technopop, from 500 to 600 nanometers of light, both don't absorb light. Okay, so both don't absorb light. I think we use another word rather than saying absorbing, because the action spectrum is not about absorption. Eh? Oh, you're saying, oh yeah, you're right. Spectrum of A and B. Yeah, so both don't absorb light. I'd mistaken your answer. Now, can we find some other similarity? We can change what he has told us slightly to get another answer. What Technopop has told us. So thank you, Technopop. Both increase to reach the peaks. I think when we say they both have peaks, that one covers that. But at what peaks do we see them? Do they have similar peaks? Or we could say they both have highlight absorption with between this and that, okay? So we look at within which regions they have highlight absorption. For example, if we look at this one, from right here to around light right there, absorption in both is high. Okay, they both have highlight absorption within that range. And then when we look at this, we look at right here, okay, and right somewhere there, absorption is also high. Okay, so within the red and blue region, someone is telling us they both have peaks. The absorption is at its maximum. Well, let's see what I have for us. So the differences, the difference and similarities. Okay, let's first talk about the differences, then we shall look at both of the answers. What differences do we notice? Any differences? Are the peaks at the same point? Someone has told us chlorophyll A has a higher peak than B. Is that true? Is that true that it has a higher peak than B at all times? Maybe that is at certain points, but it doesn't apply everywhere. Chlorophyll B absorbs light in a wider range of wavelengths. Chlorophyll B in a wider range. Okay, maybe. That is especially in this region. You can see the cursor. But when it comes this side, we see that the chlorophyll B has a narrower range. Any other difference? Any other difference? Okay, so this is what I have for us. For similarities, in both two peaks are attained. Little absorption occurs between that, one of us told us. Then the peaks are shorter, at shorter wavelengths are higher than those in a longer wavelengths. That means the shorter wavelengths we get a lot more light being absorbed than in longer wavelengths. Okay, remember, short wavelengths have more energy than long wavelengths, if you remember from your physics. Someone is telling us chlorophyll A has a lower minimum than B. Let's see whether that is right. Chlorophyll A, which is this one, has a lower minimum than chlorophyll B. That is true. They have a lower minimum. Okay. So, lower peak at short wavelength, higher peak at short wavelength. Higher peak at longer wavelength, lower peak at longer wavelength. First peak attained at shorter wavelength in blue light. Here, first peak attained at longer wavelength in the blue light. Second peak attained at longer wavelength in red light. Okay. Here, second peak is attained at shorter wavelength in red light. So you can see the differences, how they are generated if we were to look at that thing carefully. So can we explain the shape of the action spectrum? And then explain why plants appear green. How do we explain the shape of the action spectrum? Why does the action spectrum take a particular trend like that? Why does it take a particular trend? Someone is asking, what do we mean by short and long wavelength? Now, when you look at the electromagnetic spectrum, let me try to annotate. The electromagnetic spectrum, something like this. You find that it is showing us energy, assuming this is our short wavelength, wavelength, and this would be long wavelength. So in the short wavelength, you're going to find high energy radiation, such as the X-rays, the gamma rays, okay? And then as you move further this side, you find energy, you find radiations of lower energy, like maybe the TV rays, the radio rays, the such rays. So somewhere in between there, we have what we call white light. Okay. So when you put white light in a prism, okay, it is going to produce a spectrum of colors. There are 7 colors, if you remember. Red on one extreme and violet on the farthest extreme. So you find that red light corresponds to short wavelength because it is nearer to this other side. So you have red, but before red, you have what we call infrared and things like that. So, or far red. Then you have orange, then you have yellow, then you have green, then you have blue, then you have violet. So violet is a light of very low energy, but high wavelength. So from physics, there is a relationship between wavelength and energy, which is inversely proportional. So when we look at our graph here, you find that when the light is affecting the plants are able to pick out that portion of light that provides enough energy, okay? And therefore they absorb that portion of light and use it to make food. So Technopop is telling us absorption spectrum takes the shape because the rate of photosynthesis is directly proportional to energy absorbed by the chlorophyll pigments. That is correct. So you will notice that where there's much absorption, is where also the action spectrum is highest. And where there's lower absorption, that's where the action spectrum is lowest. So the action spectrum helps us to appreciate which part of light is usable by the plant to carry out photosynthesis, and which part of light is not usable by the plant to carry out photosynthesis. So that is why the action spectrum takes such a shape. So I hope that is clear. So light absorbed by chlorophyll is used for photosynthesis, like someone has said. And the rate of photosynthesis is higher in the blue and the red light regions, but lower in the green light region, because blue and red light wavelengths are more effective for photosynthesis. Okay. So the action spectrum, you'll notice, generally follows the same shape as the absorption spectrum. Okay. Except in the green light, because there are other photosynthetic pigments, such as carotenoids, that absorb some light at these wavelengths. So we are using chlorophyll A and B, but remember we also have other forms of pigments, such as the carotenoids. So the carotenoids are able to use light that is in the regions that is not absorbed by chlorophyll A and chlorophyll B. So that means that the plant is able to absorb much light, okay, because it has different pigments that have unique abilities to absorb different regions of light. So it means that if you have 3 pigments, it means that you're absorbing a wider range than if you had 1 pigment or 2 pigments. So that's why the carotenoids come in. And then lastly, why do plants appear green? Why do plants appear green? Why do plants appear green? Why aren't plants yellow or blue or red? Yes, Sandra.
So, teacher, I think plants appear green because they are able to absorb all other light spectrums, but unable to absorb green light. Therefore, reflect it, making them appear green.
Reflect heat?
No, reflect the green light.
Okay, thank you. So what we need to appreciate is that what is not absorbed is reflected. So someone is telling us that Enos is telling us that because chlorophylls absorb other colors but reflect green. Okay, not far from the answer, because they absorb light from other wavelengths but reflect green. Okay, how can we put that in a better way? So this is what I have for you. So the chlorophyll does not absorb light in the green region. I think Sandra said that. Some of us have said that. So all the other regions are absorbed, but the green one. So that means that green light is reflected. So that's why plants appear green, because light in that region is not absorbed, but rather reflected. Yeah, so I guess that's all we had for today. Thank you for your participation. I hope we are reading. This should be an eye-opener if you, in case you had stopped reading this topic in a long time. You should know that it's one of the favorite topics because there are so many questions they can set there. So you should never go for a paper when you have not read this topic. Okay. Yeah, so since we have had a very good network, as we always do, we must thank God for giving us this good network. So I want another volunteer to thank God for the network so that we can do what? Next time, we can also have a better network. Any volunteer? So thank you, those who are being thankful. You're welcome. None of us wants to pray. Shame, shame, shame. Sandra, okay. You started and you're going to close.
Okay, let's humble ourselves and pray. Gracious Father in heaven, we want to thank you for this day. We want to thank you for this is the day that you have made so that we shall rejoice and be glad in you. Father, we thank you for our discussion. We thank you for our teacher. We thank you for the good network, O Lord Jesus Christ, and we pray that you may give us the zeal to continue reading and revising. In the mighty name of Jesus Christ, we have prayed and believed. Amen.
Amen. Okay.